right, so today I am going to tell you a story about some research that I did that relates in part to the Salinas River and the surrounding watershed. And so the title for the talk I chose is called The Upside Down River, Effects of Pesticides in the Salinas River Watershed, and I'll explain what all that means. And I'm going to do my best in this talk to not use too many technical terms that you may have not heard before. So if you hear me use a $10 word that you have no idea what I'm talking about, please type it in the chat and I will explain. Or we can talk about it at the end. I'm also hoping to have time for questions at the end. But first, there are a ton of people to thank for this. And firstly, of course, Richard and Sharon, who co-advised me through this whole journey. And you heard a little bit from them at the beginning. It was a really twisty path that I took because I entered the program with some idea of what I wanted to do, but not a very point A to point B sort of direct idea of what I wanted. And so I explored a lot of different things while I was in the program. And I'm really thankful that I approached it that way, because I don't think if I would have planned it out in a linear way, I would have ended up here. And I'm really happy to be where I am now, which is to say I now have a job. And I'm very excited to tell you I am an environmental scientist with the California Department of Water Resources. So I am, I'm really happy about that. I'm excited about the projects I'm working on, and that would not have been possible had I not taken some side adventures along the way. So I want to thank Richard for being open to taking me on as a student. It was in some ways a leap of faith, which I think it is for taking on any new student, because you don't necessarily know them beforehand. And then here comes this perfect stranger walking into your office and saying, I want to work really closely with you for the next 4 to 7 years. So thank you for taking that leap of faith. And I hope that you were happy you did. I certainly am. And Sharon, that also goes, the same goes to you, even more so because we didn't even meet at the beginning. I met with Richard first, and then based on my application and on Richard's opinion, you were willing to entertain me as a student, and I really appreciate that. And also thank you to Andrea Schreier for being on my dissertation committee. I saw Andrea's here as well today. And I just thank you so much for all your feedback during that process. I think it was so helpful for me to have your thoughts and your expertise. And thank you to Dr. Xin Deng from the Department of Pesticide Regulations, as well as his staff and team. I benefited a lot from that collaboration, and also a lot of my work was funded by the Department of Pesticide Regulation, and I wanted to acknowledge that and thank them for that support, because this wouldn't be possible without them. And then, of course, other organizations, individuals have helped extremely in a lot of different ways, and I can't thank them all. I'm sure I will miss somebody, but it really is appreciated. Specifically, those of you who are willing to let me orchestrate projects with you in the lab, in the field. Again, this couldn't have happened without you. This was definitely an effort that took a lot of person power, a lot of frustrating moments, a lot of late nights in the lab or long days out in the field. And so you may recognize some faces on here. Nicole, you're at the top of the list there. Thank you so much for all your help with the behavioral analysis. Just so many people to thank. And so I just want you to know that I appreciate you and acknowledge your support through this process. And this all couldn't have happened at all without two people who are here, my parents. So thanks for having me, literally. And I appreciate that you inspired me to love nature, and that, yeah, I think that my early experiences really sort of steered me in this direction. And then I meandered on my way towards where I am now. And I really appreciate all that you did to support me in that process. My friends, my family, of course, I couldn't include everyone here, but front and center, my husband, Brian, thank you so much. I'm going to get all teary, but thanks for putting up with me for the last 7 years, and putting up with me being stressed out, hunched over my computer, late nights at the lab. I really couldn't have done it without your support through that process. And of course, my friends in dance community, who literally and figuratively have held me up through a long process. I've learned so many valuable lessons from my time with my dance community, and yeah, thank you. And lastly, I just want to acknowledge that a lot of the work that I did was conducted on lands that are traditional lands of several indigenous groups of California. And if this is something that you've never thought about or thought about for where you live, I encourage you to go to this website on the bottom here, which is native-land.ca. And they have an excellent resource available if you want to look up which indigenous groups you're standing on, literally. I think that's important for all of us to keep in mind, that we have a history that's complicated here, and we should acknowledge that. So I send my thanks to all of them. So this whole talk, everything I'm going to talk about today, which is most of the work I did in my PhD, can be summed up on one slide. However, I also have about 50 other slides that I'm going to show you to explain this slide. So if this doesn't make sense now, I'm hoping that by the end of the talk, it will. And I just really briefly wanted to kind of diverge from the science, though, and give you the human backstory. And then I'm going to give you the important concepts that you'll need to understand why I did this research. And then I'll talk about the research itself, and then tell you a little bit about why I think it matters, or why I think it's important. So first, the human backstory. So I found this picture. Actually, my mom made me this great photo album of baby pictures of myself, and I was flipping through it and I found this picture, and I think it pretty much sums up that some things change, but some things stay the same. And even though I didn't always know that I was going to go to grad school and get a PhD in ecology, I have been peering into puddles since 1984-ish by this picture. So I've always had an interest in nature, and a curiosity about the natural world. I've always loved animals. When I was a kid, I used to organize brigades of other kids to go rescue earthworms out of the gutter when it rained, because I didn't want them to drown. I used to rescue tadpoles that were in smaller puddles because those were drying out faster, and move them to the biggest puddle so that they had a better chance of survival. So some things change, but some things stay the same. However, I also didn't just stare in puddles. I had other interests. And even though the aesthetic may have changed over time, the passion was still the same. So here's a picture of me on the left in my cute little tutu, which I think I only took ballet for like a few weeks because I couldn't pay attention in class, but I really just wanted the tutu. But I did always have a passion for performance and for dance. And that has really sustained me through a lot of difficult times. I've learned so many valuable lessons through dance that I could also apply to the world of science. And some of those are being uncomfortable, but being comfortable with being uncomfortable, if that makes sense. So really not having to be good at things right away, not having to know everything right away, but being willing to stick with it until it makes sense, until you feel like you get good at it. So practice makes perfect. And I think that another important lesson I learned through this part of myself was that community matters, and community is really so valuable. And there are people that I've met through both science and dance who I feel like I will probably always know and be connected with through the rest of my life. So it was a windy path, and there were twists and turns. I went to a community college after high school, where I had some amazing teachers who inspired me in the sciences, but also in literature, arts. And then I eventually ended up at Davis as an undergraduate studying ecology and evolution. But my goal at that point was to go to vet school. I worked at some vet clinics, figured out that was a terrible idea because we ended up with 5 cats after just 3 years of vet school, or of working at the vet clinic. Ended up in a diagnostic research lab, learned about toxicology, learned that toxicology was very interesting, and eventually made it to the ecology graduate group at UC Davis, where I really felt like there was some combination of creative and technical spark that really spoke to me, and I felt like I fit there really well. And so that's a little bit of the backstory there. So it was scary to go back to grad school after having a real job for several years and having a mortgage and not necessarily having my skills as a student fresh in my mind. And I really had to use some of those skill sets that I developed through dance and through my earlier development to take that leap and not be scared and just go for it, because I knew I'd regret it if I didn't. And I really felt like in some ways this was going to be where I could find this perfect meld of things that I'm good at, things that I love, things the world needs. That was also a motivation for me. And of course, things you can get paid for, because let's just be honest, that matters too. The title for this slide, first you dig a hole and then you make a plan. When I was about 4 or 5, we had an assignment in class to describe how to build a house. And so this was my step 1 of how to build a house. First you dig a hole and then you figure out what your plan is. So I felt a little bit like that was how I started this journey of grad school. I jumped in, but then I had to make a plan. And the plan involved a lot of concepts and principles in ecology surrounding biodiversity. So here's the group participation slide that I would like you to take a moment and help me out with. What are some of your thoughts? What are some words that come into your mind when you hear the term biodiversity? This is biological diversity, and the images in this slide give you an idea, and there's no wrong answer here too. Yeah, so like new discoveries, thanks E, nice to see you. Life, yeah. Complexity, yeah, Brian, hi Brian. Abundance, everything's connected, yeah, Blake. Rainforest, beauty and richness. So we might have different words. Yes, excellent, keep going, so many species. We might have different words to describe this, this concept, but I think we all have some personal connection that we can find with this term. Complexity, oh, hi Melinda. Melinda's my boss at my new job. Thanks for coming. So yes, maybe some of these words popped into your mind. And today I'm going to talk about some ways to think about biodiversity, some ways to measure biodiversity, and why biodiversity matters in a certain system. So why does biodiversity matter? If you ask 10 different people, you might get 10 different answers. If you ask an ecologist, they may tell you that having high biodiversity has many benefits for that ecosystem. So it helps maintain the balance of life, the stability of that system, allows the system to be productive. So it produces food for different levels of organisms in the system. So everything from the slime mold to the apex predator. But if you ask someone who maybe isn't an ecologist or a biologist, they might think about it from a human perspective. So they might think about, well, I know that in the rainforest, we're always discovering new species of plants and animals that have medicinal benefits for humans. So it might be important for us to preserve this biodiversity because there's so much out there we haven't even discovered yet that might benefit humans. Also, recreation. Who doesn't love to go walk around outside in a beautiful place with plants and animals? Even if you have horrible seasonal allergies, like I'm sure many of you do, including my husband, Brian. So even despite that, you may think about, you would personally like to preserve biodiversity because you have a personal connection to it, or you enjoy it. And lastly, diversity is beautiful. So we don't necessarily need a logical reason to want to protect and preserve biodiversity other than just our own emotional connection. So not necessarily a logical connection, but it could be an emotional connection. So we can measure biodiversity in a lot of different ways. And I'm not going to go into much of this in great detail because you could take many amazing courses in ecology that can tell you much more about this than I can in an hour. But I'm just going to show you a couple of different ways that we might want to look at who's there, who is present in an ecosystem. One is looking at all the different species, so plants, animals, bacteria, all different levels of life. But that's not the only way we could measure biodiversity. We could also look at individuals in a population, looking at variation between individuals, or even looking at genes. So looking at the genetics of those individuals. So how many unique genes are there in a population? How many genetically unique individuals are there? Again, we're not going to go into that too much, but I just want to put that thought into your mind that there are lots of different ways to measure biodiversity and that biodiversity matters. Unfortunately, we also are, most of us are probably aware that there are a lot of threats to biodiversity right now. I'm not going to talk about all of these. We're just going to talk about one. But we have things like California wildfires are becoming more and more common of an issue that we have to deal with. We have drought, ocean acidification, changing weather patterns. All of these things might threaten those organisms that we care about in different environments. But today we're just going to talk about one, which is pollution, and specifically the misuse of pesticides. So I want to start out by saying that not all pesticides are necessarily or inherently bad. We benefit from using pesticides probably every day. If any of you had a salad or any kind of vegetable in your lunch today, then you probably are benefiting in some way from the use of pesticides. And the definition of a pesticide is a substance used to control pests. So this can be a lot of different types of substances. They can work in different ways. And I'll talk about a couple of things related to that. So if you could imagine the perfect pesticide, it would only affect your pest species, so only the things you're trying to get rid of off of your crop, whatever you're growing. It would be 100% effective, and after you used it, it would be gone, and there would be nothing left to linger in the environment or possibly impact anything else that you didn't want it to hurt. Unfortunately, most pesticides are not the perfect pesticide. And in some cases, we've seen in the past that we only learn this after we've been using them. So maybe you've heard of legacy chemicals or DDT, things that end up sticking around in the environment for a really long time, and also that end up hurting other organisms that aren't pests, so non-target organisms. So how are they affecting these organisms? So I'll give you a few examples here of things to think about when we're talking about the effects of pesticides. This poor stick person on the left has overindulged in many ways, and you can see that they are experiencing maybe the fact that 1 cup of coffee was great, 2 was still great, but 10 was definitely not so great. And when they combined that with their cigarette, they're not feeling so well. So the dose makes the poison. So this is an important consideration. How much of something you're exposed to can impact how it affects you. And also when you start mixing different things together, the effects can change and it can make things more complicated. So I'm not going to go into all the different details, but I want you to keep that in mind. Another important thing to think about is that pesticides might impact different species in different ways. So different strokes for different folks. So what hurts one organism might not really impact another just because of how the pesticide affects the animal and a lot of other different reasons. But these 2 characters here, we're going to talk about a lot in the next slides. These are 2 organisms that are used a lot in toxicology, a fish and an invertebrate. And the fish is the fathead minnow, Daphnia magna. We're just going to call Daphnia in the following slides. Daphnia maybe looks a little strange if you've never seen them, but they're cute little tiny water fleas. And if you look very closely into a puddle or a pond, you might see them swimming around and they kind of twitch as they swim. And that's one reason why they're called water fleas. But these are commonly used. And they're also really helpful for understanding how pesticides might affect other animals because they're sensitive to certain types of pesticides. So toxicologists, ecotoxicologists use them to help them understand the effects of a pesticide. But let's talk a little bit about other things besides just whether it kills you or not. So remember the stick figure person, he or she wasn't dead, but they definitely looked like they were not having a great day. So sometimes you can have an effect that doesn't cause the animal to die, but might still be really important to understand because it's going to affect their behavior. It's going to affect how their body functions, all the different proteins that your body is producing to function normally might be affected. So I like this, what doesn't kill you makes you weirder. I almost titled the talk that, but I thought it might have been a little much. So, okay, so this is what you've learned so far. Biodiversity is important. It's threatened by a lot of different factors, but we're just going to talk about pesticide pollution, improper use of pesticides. And pesticide effects are going to depend on how much you get exposed to, if they're alone or they're in mixtures, and also how sensitive you are to that particular chemical. And lastly, it's important that we understand how pesticides affect sensitive species in order to protect biodiversity, because if we don't think about this, we may accidentally introduce some chemical into the system that's really harmful for a lot of the organisms living there. And that would be, it's hard to undo, right? So once you have the issue present in a river or a forest, it's hard to undo that. So now I want to change gears a little bit and introduce you to where I did most of my research. If you've ever driven down the central coast of California, you've driven through this watershed, Maybe you were just going point A to point B, heading to LA or San Francisco, wherever. But as you drove through, you might have noticed that there's a lot of agricultural fields. There's a lot of agriculture occurring in this area. And it's the salad bowl of the United States. So I think it produces something like 90% of the lettuces that we consume in this greater area, not just the where I did my study, but also the surrounding Central Valley. Artichokes, strawberries, a lot of different sensitive crops are grown here. So it's really important for human food production. But it's also important for a lot of species that might be sensitive and that we were concerned about protecting, because some of these species are threatened. Their populations are declining, so they're threatened with extinction, or they might even be endangered. Some of those species only live in this area, and so it's even more important that we protect them and understand what's going on in this system. And lastly, if we want to study pesticides and their effects, it would probably be a good idea to pick somewhere where pesticides are frequently detected. So because this is an important place for food production, naturally there's a lot of pesticide use, and unfortunately sometimes that means there is some improper use and runoff. So it gets washed off of the crops, gets into the environment, and the organisms that live there have to deal with it. So here's that same map, but I'm showing you different types of habitat quality, and I'm not going to talk too much about the details and how this is calculated. This is not something I calculated, but it is a metric that I used to figure out where to do my sampling, and it's pretty well published, well described. So if anyone has specific questions, of course, I'm happy to fill you in on that. But the important take-home message for this is that there's a range of habitat quality in this area. So if you can see at the upper left corner, let's see here. Can you see my arrow? Probably not. So yes. Oh, you can, great. So up at the upper left, that's where the town of Salinas is, and that is the red circle there is showing you that habitat quality for this area is pretty bad. And this makes sense if we think about the river itself and the watershed, because there's a reason it's called the upside down river. It's because the river actually runs from south to north. So all of that runoff, all of the agricultural fields that are located upstream along the Salinas River might actually be feeding, to some extent, they're going to feed that, that runoff downstream. So that makes sense. And it also makes sense that some of the outlying areas here along the coast, this is more forested, there's less agriculture up here, would have better habitat quality. So in the really good areas, we expect to see lots of sensitive animals living there, so they don't have to worry about pesticide impacts because they aren't exposed. We see high biodiversity there, lots of different plants and animals and microbes. In the sort of bad or lower quality sites, we're not expecting to see any sensitive species, and of course lower biodiversity and fewer individuals at those sites. Okay, so let's jump into it. But first, how are we doing? Are there questions? Am I going too fast, too slow? Let me know in the chat if you need me to clarify anything. I'm happy to do them. Okay, great. So far, so good. Yeah, type your questions if they come up. Okay, so I was thinking about the impacts of pesticide and how they might affect biodiversity. I wanted to look at that in a place where I know pesticides are impacting habitat and water quality. And so I wanted to answer some specific questions related to those things. So first of all, it might be helpful to know which pesticides are present, because there are a lot of them. There are hundreds and hundreds of different chemicals that are commonly used for different purposes, and at different times of year, different crops. So understanding what kinds of chemicals are present is going to be really helpful for the next questions, for answering the next questions. Next question, is the river water toxic? And I put a little asterisk there because when I first was thinking about toxic, I was thinking about, does it kill them or not? So that toxic, I'm just thinking about, do they survive? If they're exposed to this water, do sensitive species live or do they die? But then also working up from there, maybe it doesn't kill them, but like you remember from the stick figure, maybe they don't feel so good after they were exposed. And maybe that does have implications for their survival later on, or reproduction, or other things that are important for those species. So the third question is delving a little more deeply into how those effects, or what those effects are, even if they're not just killing them or not. So in order to answer these questions, I was lucky enough to partner with the Department of Pesticide Regulations, who does a ton of routine chemical monitoring in this area, which is another reason why this is a great place to study, because we have all this historic data about chemicals that are detected in this area. So I went out with them, I collected water samples, and brought those water samples back to the lab. Meanwhile, the Department of Pesticide Regulations conducted analytical chemistry on the samples to tell us exactly what was in the water. So back at the lab, we exposed our fish and our Daphnia to this water. And then we looked at a couple of different things. We looked at, first, did they die or not? Second, what was their behavior like? So how did exposure to this dirty water affect their swimming behavior? So the center picture is a little box that has a camera mounted at the top, and the slide on the right, you're gonna see, is what the camera records. So the camera is recording all these little tiny fish, and they're all inside of a plastic dish that has little cups inside of it. So it's a plate. And they all are in a little bit of that water from Salinas. And we recorded them and watched how their movements changed, and whether their movements were different in ways that might tell us something about how they were responding to exposure to this water. Oops. There we go. So I'm not going to tell you too much about the analysis and the analysis for this part, or for the bioinformatics for the second part I'll talk about. Needless to say, none of you want to hear about that today, because today is a story about what happened in the Salinas River, not a technical talk. However, I will tell you that it was a lot of work and a process, and I'd be happy to talk to you about those specifics after this talk if you're interested. There were definitely moments where I felt like this character in the bottom left, if you're not familiar with him already, who realizes that he's in danger because there was definitely there were frustrating moments during that process, as Nicole can tell you, because she helped me a lot with that. All right, so recap, what's in the water? How did we answer that? The Department of Pesticide did chemical analyses. So let's look at what they found. I'm going to show you some real data. It's like one of the only slides that has numbers on it. But first I want to tell you a little bit about what it means. So it's color coded. On the left side, you're going to see the darker red color means it's more toxic to fish. The yellow and then the lighter yellow indicates that we know from previous experiments that that level of a chemical is going to negatively impact our Daphnia. And then if there isn't a color in the table in that box, then we're pretty good. It's below EPA thresholds, which means this is the Environmental Protection Agency. They set thresholds that they've decided are safe for these animals. So you don't need to worry about the numbers. But if you look at the colors from 3 different sites, you can see that we have some big differences. So site 1, we've got several chemicals present at levels that are known to negatively impact fish, sensitive fish, as well as chemicals that are negatively impacting our Daphnia. Site 2, still a lot of Daphnia impacts, not any necessarily high enough levels to affect fish. And then at the 3rd site, ND means non-detect, so it just means that there wasn't enough to be detected, or there was none. And so there's a few there, but not as bad. And what I'm showing you here are only the chemicals that were detected above a threshold value in 1 or more sites. So these are not all the chemicals that we found. These are just the ones that we focused on because they exceeded some value that might mean they were hurting the fish or the Daphnia. So how did they affect survival? So based on this slide, what do you think? Site 1, you think the fish are gonna survive or not? We can take bets in the chat. Maybe not. Yeah, it's bad news for fish, maybe. Maybe. Okay, awesome. And then site 2, Daphnia, probably bad news for Daphnia for site 2. Maybe the fish are okay. Yeah, and then site 3, not so bad. There's a couple of things there, but okay, it's at the lower end, so maybe we'd be okay. So let's look at survival here. But first, I'll remind you, or just maybe have a little caveat about this, that we did multiple sampling events, and the chemistry that we found depended on the site we sampled and when we sampled. So that graph that I just showed you, that's 1 sampling event for 3 sites, but that is just a snapshot of what we found. So there were other sampling events where we saw different chemicals, different concentrations of chemicals, different numbers of overall chemicals. But what we did find was that pesticides, at least a few, were detected at all sites. This is not surprising because the Department of Pesticide Regulations chooses these sites because they expect to find things there. That's why they want to monitor them. So remember on that map, there were those red sites that were expected to have bad habitat quality. That's where these samples were taken. And lastly, even though there were a lot of different pesticides, most of them were pretty low levels. So not all of them exceeded any value. So with that in mind, and with our guess that we had earlier, let's see what actually happened. Oh no, this is expected. Okay, this is to remind you what we expected. This is not what actually happened. So we expected that first site, not so good for fish, but the other 2 sites, okay for fish. The first site and the second site, probably not so good for Daphnia, but the 3rd one, we think they're gonna be okay. Okay, let's see what happened. So what happened was we had pretty much 0 mortality. So all the fish survived in all the sites. And we had very low survival in Daphnia, even when we took that site water and we diluted it down. So it was mixed with clean water. So they got less toxic water, more clean water. We still had mortality even in that lower site, although not as much with the site 3. It was a little less. That's why I only put 1 of the red X's. So some of them survived. So what does that mean? So if we only looked at chemistry, we would have learned some valuable things. We would have looked at the fact that there are complex mixtures of chemicals in most of these sites. We already know from previous studies from the literature that Daphnia and fish are sensitive to some of these pesticides. And the pesticides we found are at levels that we think could affect them. So they could kill or hurt the Daphnia or the fish. When we looked at chemistry plus survival, we saw a little bit of a different picture, which was that fish actually did better than we thought they would, but maybe Daphnia didn't do so well. But this doesn't give us the whole picture because it's only looking at whether it killed them or not. And I'm going to remind you again of that sad, poor stick figure who's drinking his too many cups of coffee, smoking too many cigarettes. And so now we need to think about how else this water might be affecting these species. Are we good so far? Does anyone have a burning question? Okay. We're good. Great. Okay. So what doesn't kill you makes you weirder. So we wanted to look at how exposure to these different water samples might affect the behaviors of fish and Daphnia. And in order to do that, I showed you the slide of the video chamber, and we're taking videos. We did something else too. We turned the light on and off. And that is something that is important to look at for these organisms for a lot of different reasons, but it's a stimulus. So if they see a shadow go over them, for example, that could mean a predator is nearby. And it would probably be a good idea for them to run, hide, do whatever avoidance tactics they're capable of doing, right? Just get out of the way. So if we change the light, that is going to simulate some sort of stimulus that might have a similar impact in the natural environment. Okay, the second real data slide that I'll show you. So on the y-axis, so when the lines move up and down, that is looking at how far and how fast they moved. And then we have over time, so moving forward in time from left to right. And you can see that we started them out in the dark, and then we turned on the light, turned off the light, turned on the light, turned off the light. And everybody seems to react to that change in light conditions, whether they were exposed to water from site 1, 2, 3, or if they were in our control group, which is in blue here. So the controls are what we would expect them to do normally without any pesticide exposure. So there's some similar patterns here, but I think what you're going to notice is on the right side, that water from the two sites with the more chemicals and the higher detections starts to have a really different baseline level of movement. So those fish are moving faster and a little more erratically than the other groups. So that's interesting. So they're hyperactive, but just in the sites that have more chemicals, right? And they also had a decrease in how much they were able to respond to that light and dark stimulus. So they didn't freeze when the light turned on quite as much as the other ones did. So now we have a lot of information now. And we've based our findings off of several different testing methods. So we started with the chemistry, super helpful. We saw mixtures, we saw what was there. We added survival, did they survive or not? Yeah, the fish did. Daphnia, it's pretty clear that this water negatively affects them because they pretty much all died. But then with the behavior, we saw that the fish were acting kind of weird in the water from the sites that were more toxic. So even though it didn't kill them, we are seeing that there are some behavioral differences for those groups. So lastly, we wanted to look at what's going on. And because these samples have a lot of different chemicals in them, and we're not able to focus on one chemical and its effects on the fish, we can't say for sure which chemical or which combination of chemicals is causing these effects. But we can say that exposure to this water overall is having some interesting effects, not just on their behavior, but also inside the cells of the fish. So we saw that fish were producing more of a detoxification enzyme. I couldn't find a good image to use for this, so that's like a green smoothie. So it's like a detox smoothie for you. If you're wondering why I put a green smoothie on the slide, detox. It doesn't actually work that way, but that was the closest image I could find. So we saw that they're producing more of this enzyme that helps them break down chemicals and process them out of their body, some chemicals. We also saw that they were not producing the same levels of something that helps them control their neuromuscular functions. And it's related to the calcium channel, voltage-gated calcium channel. But basically, it just interferes with their ability to make decisions, possibly, and execute them, possibly. And if you want to read all about it, you can in this sort of hot off the press publication of this part of the data, which came out, yes, it was in December. Yeah, December. And I put this picture of me Christmas morning because I'm so excited that it finally got published. It was a lot of work. It was a lot of effort by all the authors listed here at the top. So if you're interested in the technical details about the gene expression and some additional studies that were done by one of the co-authors, then you can find that, or I'd be happy to send it to you as well. All right, so we looked at what was in the water. We looked at how toxic it was. We looked at other effects that it has on fish and Daphnia. And things that were useful and we learned from this, that I learned from this, was that using multiple approaches is giving us a more complete picture of the toxicology that's occurring in these environments. It's useful for understanding how complex mixtures of pesticides are affecting these sensitive organisms. And that's important because, as you saw from the chemistry, there were often many different pesticides present at each site. It's also helpful because we used species that have extensive literature. So there are a ton of papers published that use these species. So we had a lot to compare it to. But what about the other animals and plants and organisms that are living in the Salinas River watershed? So obviously these two are not necessarily the only ones or relevant for this system, right? These are what you call model species. So they're good to model effects, but they're not necessarily representing what's occurring in the natural environment. So we're gonna add a last question. We're almost done. And we wanted to know, okay, well, in order to understand how organisms are maybe getting hurt by water quality, we need to know who's there. So how do you know who's there? So I showed you this slide that there are good and bad habitat quality. This is using something called the California Stream Condition Index, which is just one method, but several methods will use a traditional monitoring technique, biomonitoring. You go out, you take a net, you sample a bunch of bugs and other little critters, and you look to see who's there. And if you see a lot of very sensitive organisms, then you can say something about what you would infer the water quality or the habitat quality to be like. Obviously, in some places, we wouldn't expect to see those really sensitive guys, especially this ditch by the side of Highway 101, which was one of the sites that I had the privilege to go to. It's literally like you have to pull off on the freeway. Like you look at Google Maps and hopefully someone else is driving and you're looking at it, you say, okay, now pull over now. And then you get out of the car and hope that the CHP doesn't wonder why you're there. So how do we want to look at all these different sensitive species that are there? That's kind of overwhelming, right? Because if we do the technique I showed you, where we sample with nets. We have to go back and physically identify all of those animals under the microscope. I'm not a taxonomist. I'll leave that to Sharon. But I know from my limited experience, it's really hard to do, and it takes a lot of expertise. So another approach that's being developed is using DNA that's shed by all these organisms into their environment. And that is abbreviated as environmental DNA. How does it get there? You guessed it. Actually, many different ways, but organisms are constantly shedding DNA into their environment. Some shed more than others. Maybe an adult organism sheds more than an egg, but you get the idea that it's there, it's out there, and if we can figure out how to collect it and test it, we can, in theory, figure out who's there. So I went to all these different sites and I collected water and sediment, and with the help of my #1 field assistant, my husband Brian, who went with me on a lot of those adventures and who was extremely helpful through the process. Here he is on the left. We had a filtration system for our water samples that used a drill as the motor. So we could filter the water, freeze the filter, and then take that frozen sample back to the lab because it was better preserved that way. For some of the sites, I also did the traditional method of collecting the bugs. And I'm going to talk about that a little bit. Oh, and then the middle tube here is a sediment sample. So soil, but at the edge or in the stream. So what did we find? So I looked at both methods. With the help of an amazingly talented taxonomist, Priscilla. Thank you, Priscilla, for all the countless hours of work. We would come in on the weekends and listen to podcasts, and I would help her with this. So we identified just from I'm going to show you data from one of the sites, not from all of them. But we identified a lot of different bugs. And then we looked at, are we seeing the same bugs in our DNA samples, or do we see different things? And overall, we found a really good overlap with what we found in both methods. And there's a lot of reasons why there isn't a perfect match that don't necessarily have to do with what organisms are actually present there, but I'm not going to go into that in too much detail. But needless to say, it was a good sign that we saw overlap. We also saw that in the sites with better water quality or habitat quality, we saw more sensitive species and a few in the okay sites as well, but none in the really bad sites. So that site I showed you that was the ditch by Highway 101, no bugs there, no sensitive bugs. We also found that in the sites with better habitat quality, we had a higher total number of species, so higher biodiversity. That makes sense, right? It's better habitat quality, better water quality. So just to recap that last part, we saw that sensitive species are present only in those better, the really good to the okay sites. Good sites had higher biodiversity. And also the biodiversity was really different between sites. So we could actually differentiate the site by what organisms we detected with this eDNA method. All right, so we got similar results. And this is this is like the future hope that this monitoring technique can evolve to. So it would be really nice if we could go out and we could take our little handheld meter and we could point it at the stream or take a water sample, whatever, and it could tell us in real time what organisms are there, because it would save us a lot of time and effort, a lot of resources to be able to do it this way. So that is the Star Trek tricorder future scenario, but it's getting closer and closer. And it's all part of this idea of molecular biomonitoring. So monitoring how impacted or how pristine a certain place is using these molecular tools. And with that, so that kind of wraps up what I showed you at the beginning, which is this overview slide of all the different research projects that I talked about. And hopefully you can see how they all sort of fit together in terms of improving how we are able to monitor how impacted these sites are. And so hopefully you're all still awake and here, and I appreciate your attention. Thank you so much for being here and for all your support throughout this whole process. I'm happy to answer questions. And also, if you happen to be in the Sacramento area, we're going to have a little celebration tonight. But if you're not, then we'll have another one some other time, no pressure. But just to put it out there that we will be at Sac Yard on 33rd Street.
Hey, well done, Sarah.
Thank you.
That was a really clear presentation, and I love the way you tied everything together and just had that nice thread going through everything. So very, very well done. Very proud of you.
Thank you. And it's funny because when you talk about it at the end, you can make it sound like it all went really smoothly and linearly. And of course, that wasn't really the way that it all happened, but it was nice to have this opportunity to think about the story and to think about how everything fit together and how to tell the story, because humans like stories. So I hope you got the story and weren't too bored with the details.
That was cool. One of the things that you've always excelled at is science communication, Sarah. So very well done. So with that, questions for Sarah, or compliments, or bring it on.
Yeah, okay. I see some hands. Maybe if you can use the raise hand function or type in the chat so then we don't have people talking over each other. Euphoria, I see your hand first. So I'm going to, can you unmute yourself? You can unmute yourself, right? Yeah.
Yeah. I was really interested in the locations of your sites. Oh, there's my camera. Can you show the slide again with the map that had the sites listed? It was just so brief. I'd love to look at that for a sec.
Yes, let me go back to that. Let's see here. Hold on, I have two monitors, so I have to switch over. Can I do that? Okay, so here is a Google.
Oh, yes.
Yeah, was that the one?
Yeah, so your sites 1, 2, and 3, those are on those top 6 dots somewhere in there.
Exactly. Yeah. So these are sites that the Department of Pesticide Regulations has monitored for more than a decade. And so they have extensive chemical data from these areas. And like I tried to explain, they expect to find chemicals there, pesticides there, because it's part of why they monitor them. There's a high likelihood they would be there. And Shin, I see your hand up. So I'm going to call on you next because Dr. Shin Deng is from the Department of Pesticide Regulations, so maybe he wants to add something.
And can I just ask one more question?
Oh, sorry.
Here?
Yeah.
Where you're sitting in the sampling, is that one of the sites with a lot of pesticides?
No. So this site was actually Shalone Creek, which is in Pinnacles National Monument. Most of the sites actually had much more water flow. This was a site that we debated, or I debated excluding, because we didn't have a lot of flow at the site when Brian and I ended up going there. So we were concerned that the water temperature was going to be high and we wouldn't see as much diversity. But it's a national monument. It's more pristine, less impacted. So we were hoping to include it as a reference site. Yeah, so it was beautiful up there, but there wasn't as much water as we were hoping for.
Cool, thanks.
Yeah.
Super interesting, thank you.
Thanks. Okay, Shin, go ahead.
Hey, well, great talk. I enjoyed it. And, I was expecting to see a lot of figures and graphs, tables. This is great. Yeah, I was wondering how you're going to craft all this stuff in 1 hour. Yeah, congratulations I experienced some of your up and downs over the years, and see the path you get here to get Dr. Stinson to be Dr. Stinson, and thank you for the hard work you have done over the years. I have managed many contracts, many projects through our program. This is by far the most interesting, well, because I'm a aquatic toxicologist and you are. So, and also I think it's the most productive project we had so far. So certainly money well spent for our program.
Thank you.
So I really don't have any questions, technical questions regarding the projects. And we have go through of them, many times. But I am interested to know what's your thought about the potential projects, or what are the ideas you have for the future moving forward.
Yeah, I think that, thank you, first of all, for that. I really appreciate your support, as I said at the beginning, and I had a lot of fun putting this talk together. My goal was to make it accessible for everyone in the audience, so I tried not to make it too technical. But my thoughts about ideas for projects moving forward, I do feel like there is a lot of potential with environmental DNA monitoring, as you've heard me go on about at length, and that I convinced you to support this project with that idea. And I think now that it's becoming more prevalent and more agencies and universities and other groups are starting to implement this as a monitoring tool, I think one important consideration moving forward is to make sure that we're all standardizing our methods as much as possible and kind of communicating with each other. So again, back to like Richard was talking about science communication, I feel like, yes, we need more on the ground research to understand how to use that as a monitoring tool, but we also need more analysis, development of pipelines for analysis, communication between different groups, thinking about data storage and data handling, cleaning the data, all these little steps that happen after you sequence your DNA. Those things can have big impacts on how you interpret your findings, and sometimes I feel like that part gets glossed over, and maybe people who get excited about DNA, eDNA, don't have a background in it like I didn't. And it's easy to make mistakes or to not think about how what you do can impact your analysis and your results. So I think more studies looking at analysis pipelines, statistical methods, in addition to obviously like going out in the field and sampling and trying out new methods. Do we use water filtrate? Do we use sediment? All those sorts of logistical questions are important too.
Well, one more question. So what are you going to do, when you work with water resources? Now you're going to continue with your work?
Yeah.
Or maybe we can collaborate in the future. Now you're part of the state workers.
Well, the group that I'm in is collaboration, science, and innovation. So there's collaboration right in the title, but it's actually a new genetic monitoring lab that DWR is starting up. So we're specifically focusing on how the State Water Project might impact listed species. So we're not going to do all the different types of monitoring, like Fish and Wildlife does other genetic monitoring as well. So we'll mostly be focused on that and meeting the ITP mandate for that. But we want to collaborate and we want to share and do projects with different groups. So that is something that's high on our list. So yeah, Sherlock, Richard dropped the Sherlock reference. So if you don't know what Sherlock is, that's a talk for another day. But it is a really cool new technique that Melinda Bearwald, Daphne Gill, and others are developing and innovating, and Andrea Schreier and others, that uses a CRISPR gene editing technique to look at environmental DNA. So it's pretty cool.
Cool. Keep in touch.
Yeah, I will. Thanks so much for joining. And
Okay.
I see E. I saw did you have your hand up? Yeah, E, go for it.
Hey, Dr. Stinson, how are you doing?
I'm
My name, for everyone that doesn't know me, my name's E. Nunez. I use they, them, theirs pronouns, and I had the absolute pleasure of supervising Sarah as a graduate assistant in the undergraduate research center on campus. And at the undergraduate research center, we try to encourage and motivate undergraduate students to take on research experiences to hopefully grow up and be a Dr. Stinson one day. And I just want to say, Sarah, it has been an absolute honor to work with you, to be next to you during this journey. I always knew that when it rained, there were water samples that had to be collected. So I knew that that was a day where things needed to be flexible in the office. And there were days when you came in and you were stressed out and working on your dissertation or your research, and you were still available for our students and supporting our undergraduate students. So from our URC team, congratulations. We are so proud of you. And we are so glad that you have impacted our team for the better and have supported the undergraduate research enterprise on our campus. And we can't wait to bring you back and as an alumni to talk to our students about undergraduate research. So thank you, thank you, thank you for always coming back to our team. We've created positions to keep Sarah on our team because she is so amazing. So congratulations, Sarah. We're so proud of you, and thank you for an amazing talk.
Thank you so much, E. I really appreciate that. And it's been my pleasure to work with the URC too, because I think what everyone does there is amazing. And it's really great to see students come in who don't know what they want necessarily and who are trying to figure it out. And I can definitely relate to that, being in that place and feeling a lot of pressure to figure out what your career path is. And usually they're amazing and they have so much potential, and they just need someone to tell them not to be scared and just do it. So thanks for that experience. Okay, I see Kyle.
Hi, congrats
Hi.
congrats and excellent talk.
Thank you. I didn't recognize you with your luscious lock.
That's probably brilliant. So I had a quick question about eDNA. I imagine a challenge with working with eDNA in rivers is that eDNA will go downstream and potentially affect sites downstream. Did you encounter any of that? And how did you get around that to figure out what was going on?
Yeah, that is a great question. And I did think about that a lot. So there is a paper called Rivers or Conveyor Belts of DNA, or I'm misquoting it, but basically that is a concern in the field, because when you're taking a water sample, the water's flowing downstream, you're probably getting all the DNA from upstream, like you said. So one of the reasons I chose to focus on sediment samples was that there was some literature that suggested that you're getting a better picture of local biodiversity from sediment. So I chose riffle sites, and I selected sediment that was immediately downstream of riffles. And that was a place where I might have expected to see more diverse, more sensitive species. And so you can't get around it completely, but my hope was that it was an improvement. And what I found when I compared the water to the sediment, more fish in the water samples, so more fish detections, more invertebrate detections in the sediment, but there wasn't a complete overlap again with the species detected. So that was interesting.
That's very cool. And I think it's also fair to say that the inverts themselves are also going to the invertebrates are also going to wash downstream as
Yeah.
well. So that's another thing also going on. Very cool. Thanks for sharing.
Thanks. Oh, and sorry, Euphoria riffle. So shallow oxygenated, fast moving water where you might expect to see more invertebrates that need oxygen and need better water quality in general. So it's not like stagnant, still deep pools. It's more the shallow, rocky parts. Oh, Nona, I see your hands up.
Hi, nice
Hi.
job. Very
Thank
nice,
you.
very impressive. So, you know, obviously when you start something like this, you have some idea of what you're looking for. What were your biggest surprises besides how much sleep you went without?
My biggest surprises. How annoying bioinformatics can be when it doesn't work. No, I think my biggest surprises were, but related to that thought, you have to be very careful about how you clean your data. So when you go through and you get all this raw information, right? And you have to decide what is a real signal, what is really there, and maybe what is the result of contamination, or maybe when you sequence the DNA, there was a little error that happened and you swapped one thing for another thing, and now it looks like it's something else, right? So you have to be careful about that. And an example of that is I got just one sequence, which normally you would discard things where you only find one sequence because it might be an error. Rhinoceros. So that was a surprise. I don't think it was real. I think it was just a kind of a sequencing error possibly, but you never know. And it could be that there's some safari wild animal park upstream. Yeah.
Wow.
Someone might have a pet rhino. I know my parents were looking at a property to buy, and I sent them this property that was right near the what's it called? The Safari West. And they literally would have had they bought this place, like the deck looks out over the rhinoceros enclosure.
Wow. Oh, that's awesome.
Yeah. So yeah, if there's a rhino wandering around Salinas Shin, please let me know.
Yeah, dang. Well, very impressive. We are very proud, very impressed.
Thank you. Let's see. Oh, Florian, I saw Florian's comment in the chat. Is it possible to conduct longitudinal site monitoring, confidence range of biodiversity in which a site is considered as normal? Yeah, I mean, in theory, there's a lot of variables in that region, and unfortunately, you transition from highly impacted agricultural areas into surrounding national forest land pretty quickly. So you have a lot of diverse sites and challenges associated with those sites. So I guess, yeah, you could. I mean, it's possible.
Did you sample these sites at different times?
Did I sample the sites at different times? Yes. So I couldn't go to all the sites at once, but
but sorry, what I meant was like several times each site.
Oh, multiple sampling events? No. And
Okay.
what might be an interesting study would be to resample the same site over time, or same sites over time, because you do have a lot of seasonal variation in the types of specifically bugs, macroinvertebrates, so big bugs, that you would see over the course of a season because they're hatching out, they're developing in the streams or rivers, and then some of those as adults, they actually emerge and they're flying insects. So you might see a signal for when some of those emerge and hatch that might be useful for understanding conditions in the stream. So yeah, that would be great. It's my next PhD. I can do that.
Cool.
No more. Other questions I can answer for people? I think that's it. Yay, we did it.
Yep.
Oh, Sharon.
Well, I just wanted to quickly thank E for their comment, because Sarah was also an awesome TA in my class, and I meant to mention that earlier, but yeah, just awesome all around.
Thanks, Sharon. I appreciate that. Yeah, and let's go get a beer and celebrate. Suzanne's in, I can tell. Thank you. Thanks, everybody. I really appreciate it. And I look forward to hanging out with you and doing cool science, dance, whatever it is in the future. Thanks for showing up.